I want to welcome everybody to the meeting. I want to thank the presenters for giving me the opportunity to talk to you all, and I hope that I justify their trust in letting me talk first. I'm going to talk about hospital-acquired infections. Quick disclosure, ONT did pay for me to travel to the meeting. My other disclosure is that I'm an MD. I don't do a lot under the hood in terms of sequencing, and so if you ask me extremely detailed questions about ONT, I'm going to frown a little bit, but I'm going to try to keep this kind of clinically oriented. So what I'm going to talk about today, I'm going to talk about why we're interested in tracking bacterial pathogens in the healthcare setting. We heard a little bit from Gordon about how ONT is being used from an infectious disease standpoint. I'm going to expand a little bit upon that. I'm going to talk about our project. That'll be the nuts and bolts about using Oxford Nanopore sequencing to assess potential transmission of these pathogens. And I'm going to touch briefly upon future directions. So I realize not everybody in the audience is a microbiologist. I don't find fault with you all for that. But healthcare-associated infections are a really important cause of death in the United States. So what is a healthcare-associated infection? So I work at a cancer center, and let's say a patient comes in to get chemotherapy. 4 days later, they're in the hospital. They develop a bloodstream infection, in this case with E. coli. We would call that a healthcare-associated infection. And the standards of how these are defined are set by the National Healthcare Safety Network, or NHSN. And there is a lot of these infections. I just put up the data for the United States, but go around the world. They're a huge problem. When you get into places that don't have vigorous infection control operations, they're even more of a problem. So again, a huge issue clinically. As we've gotten more complex with how we integrate healthcare into patients' lives, it's becoming increasingly difficult to track these pathogens. So in the modern healthcare system, such as MD Anderson, really complex patients are getting admitted in the multiple times. They're getting treated in outpatient settings, and the time from being exposed to one of these drug-resistant bacteria until you manifest disease can take weeks to even months. And so that makes this tracing really difficult for standard infection control practices. So for example, they might say, did the same patient or multiple patients develop infection at the same time on the same floor? And that actually really rarely happens, as I'll show you. So how does typical infection prevention and control work in America? And this is true basically of all, like Western Europe as well. Basically, you've got baseline rates for particular infections, and then they look to say, oh, we had more than this kind of baseline rate. Maybe we had a cluster of disease in one area, and this might trigger an investigation. We don't routinely do sequencing on every pathogen. The investigations might include some type of molecular characterization, and then the transmission interventions can be very serious. It could be as simple as removal of, say, a point source, like you have a contaminated endoscope from GI procedures or a sink drain. But when I was a fellow, we had a huge outbreak of Acinetobacter in our public hospital that had an ICU, and they had to eventually close down the entire ICU and rebuild it from scratch. So tens of millions of dollars can be at stake, in addition, of course, to the impact on patients' lives. When we've done a lot of sequencing at MD Anderson, what we notice time and time again is that there appears to be cryptic transmission of bacterial strains going along. So what do I mean by that? Well, we take a lot of strains, we sample large cohorts collected over many years to try and analyze what's the molecular epidemiology, and time again, we would see clusters of bacteria that were really highly related. And so I put just some examples on this. Linezolid-resistant Staphylococcus epidermidis, where they might differ by 0 or 1 SNP across many patients. Drug-resistant Escherichia coli or Klebsiella pneumoniae, and it wasn't just my lab that was seeing this. My boss, Dr. Roy Shamali, saw this with VRE. So we kept seeing these clusters of transmissions, but these studies were done years after the infections. So here's an example of a paper that we published in 2024. On the left are a couple of clusters of particular types of ESBL Klebsiella pneumoniae. The blue bars are individual patients that represents their time in the MD Anderson system, and the red bar represents highly related, genetically almost identical organisms. And you can see by the x-axis, this is happening over years. So we're seeing identical isolates that are popping up in our patients, causing life-threatening infections, but some of them are as long as 4 or 5 years apart. And when we go to infection control and say, can you investigate this? They're like, what are you talking about? These patients were in the hospital several years apart. How do we even begin to investigate that? So what are the challenges for using whole genome sequencing to investigate transmission of these strains? Well, we want to basically acknowledge that we follow in the footsteps of the University of Pittsburgh, especially Alex Sunderland, who has published a really pivotal study in 2022 where they basically sequenced everything that had come in the hospital for 3 years, about 3,000 strains, using the Illumina platform, and they found about 10% of their isolates were genetically related, and critically, less than half met those kind of classic criteria. So over half are having cryptic transmission. And I'm not going to show you every paper that's come out in the last couple of years, but just to say that this is not just happening in the US, paper in Australia showing about 30%, and again, using the Illumina platform. But a critical thing is this time of reporting. So about 70 days it took from the time the bacteria was isolated until they got back to the clinical service saying that, hey, you've got a transmission event going on. That's way too long. Nobody can act in that kind of period to interrupt transmission. So the summary as we move into our project is whole genome sequencing shows that a lot of healthcare transmission of pathogens is going on, it's going undetected, and it's being identified often months, or in our case, years later. So we wanted to try to move that into a much more prospective idea. So what were the major hurdles to try to identify these things in a timely fashion? Of course, time. Can we be able to provide the infection control people with rapid, accurate information? Money, can we do it in a cost-effective way? If you're going to add, say, $100 per sequencing to every bacteria, that's considered kind of low cost these days, but that's going to add thousands of dollars to your microbiology budget, and very quickly your project is going to get shut down. Complexity of bioinformatics, of course, trying to analyze on the back end, are strains related? What are they related to? And of course, how do you integrate that data into workflows of infection control? So how the project I'm going to tell you about today begins, it begins by me being ignorant. As usual, I was approached by a master's student who had a previous background in infection control, and she wanted to do her master's project about using Oxford Nanopore sequencing to detect hospital transmission. So I peeked my head up and said, that doesn't work. We all know the error rate is too high. We only use Oxford Nanopore to supplement our Illumina sequencing, and so, why are you bothering me? She said I should read the literature more, that in 2022, the Oxford Nanopore 10.4 data had come out showing that you could use Oxford Nanopore in a standalone sequencing project. So I kind of perked my head up a little more, said, but I don't have any mechanism to do this right now. My NIH funding is dedicated to other things. And I said, you're going to have to do the project by yourself. I don't have laboratory personnel to do it. And I assumed that being sequencing, it was really labor intensive. And she said, nope, I can do it. So everything that was done by her, which is really quite a remarkable accomplishment. So what did she do? This is the overview of the project. I'm going to go through step by step in the next couple of slides. But basically, she went down to the microbiology lab and to grab plates that had target pathogens. I'm going to talk about why we specialized or focused on a couple of target pathogens. We assessed the likelihood of transmission clinically. She and I would review charts every couple of days. And she sequenced about 10 isolates a week using ONT sequencing. I'm going to go into a little bit more detail. She then measured the genetic relatedness between each of the bacteria to look for evidence of transmission. And then she combined that with epidemiologic data by our collaborators in the infection control department at MD Anderson. Okay, so getting back to our debt to the University of Pittsburgh, how did we target our process, right? They sequenced every single bacteria. That's really not feasible for one graduate student to do. So we went back to their paper and said, which of these strains are actually being transmitted? And again, I'm not going to delve too much into it, but there's certain strains where you'll see zeros next to them, like these strains are never transmitted in the hospital, and others that reach 10, 15%, like drug-resistant E. coli or vancomycin-resistant enterococci, which are typical organisms causing these kinds of infections. So we said, okay, let's get to a subset of strains that you could do in a fashion that are considered high risk. The final list is listed up here. MRSA refers to methicillin-resistant Staph aureus. That is common enough that even some of my patients know about that. VRE refers to vancomycin-resistant enterococci, and then carbapenems are kind of last-ditch antibiotics. So these are kind of the worst bacteria that you can find in the hospital. That's what we're going to sequence. So what were the goals of the project? Really, the primary goal was for her to develop an Oxford Nanopore sequencing pipeline that was fast. We wanted to get the data less than 72 hours from starting to work on the isolate to completing the genetic analysis and getting that information to IPC, right? So we're trying to move from 70 days to 72 hours. It had to be accurate. The error rate had to be lower than the standard threshold for assessing bacterial relatedness, which, as I'm going to show you, is pretty tight. So the error rates had to be really low. And it had to be low resource, okay? I'm poor. She was a single graduate student. She was using MinION flow cells, not in a sequencing facility. The secondary goal was to determine if any of these pathogens were being transmitted at our hospital, similar to what had been seen. So here's an overview of the project. It went on for 26 weeks. She had to take a month off to go back to where she lived, but overall it's a 26-week project. Her and I screened 494 isolates, and 246 of those met our criteria. So what were we looking for? Things that caused infection in the hospital, or say the patient just had a surgery, or other integrated aspects of our medical system. If the patient was transferred from another hospital in septic shock, we weren't going to look at that isolate, right? We're looking for stuff that might be transmitted in our hospital. So about half of the isolates met our criteria for possible transmission. She ended up sequencing 246, 1 failed sequencing, and 3 were identified as different species relative to what the clinical microbiology lab told us could be an isolate error or could be an error on the automated system on their part. We threw those out. So we ended up with 242 strains, or 98% met our criteria, and the breakdown is shown there. About a third of them were MRSA, and then a scattering of other bacteria. Okay, so here's the part that I had to put in. My computational biology people went over it, so I hope I don't make too many errors, so I apologize. The genomic DNA was all done by her. She did using Genolutes directly from the clinical microbiology samples. So basically, she went to the micro lab, got the plates, scraped the plates, isolated the DNA. She used the rapid barcoding kit, and then she performed sequencing using the R10.1 Flow Cells using the MinION sequencer. Originally, as we started doing the base calling with Guppy. This changed to Dorado about 1/3 of the way through the project, thank goodness, in super high accuracy mode. And then Will Shropshire, who is the computational biology person in my lab, has basically a GitHub site where he uses this thing called Flyest that allows the sequencing consensus assemblies to be made. So here's a summary of the data. Again, 245 samples are included here because I hadn't thrown out the 3 yet. The couple things I really want to pay attention to, the coverage depth was somewhere in the 80 or so range, or the coverage median was about 80. But really, really critically is we got very high quality data for analyzing our genetic targets. So that's the yellow line, 99% plus ability to analyze our targets, which is well above what's needed to call genetic relatedness among bacteria. So how well, in addition to that, did the sequencing work? Okay, being a sort of, not a sequencing guru, I'm going to say much better than I expected. Time, 24 hours from when she grabbed those isolates to when she completed genetic analysis. She averaged 21 strains sequenced per MinION flow cell. Being cheap, we reuse the flow cells, usually 2x to 3x. So she would sequence 4 or 5 hours, stop, wash, sequence 4 or 5 hours, stop, wash again. And the output was around 20 gigabases, again, with reuse. We met our sequencing QC to proceed, again, 245 out of 246 samples, so it really worked well. And then the accuracy, I'm going to go over the next 2 slides, critical. We took the same genomic DNA and did Illumina sequencing for about 50 or 60 or so of the samples. We then mapped the Illumina reads to the consensus assembly for the ONT and said, if there's errors, it's an ONT error. So that's how we were assessing accuracy. So as I said, really, really helpful. When we switched from Guppy to Dorado, our error rates basically disappeared. What I'm showing here are the number of variants on the y-axis. On the x-axis is in red are single nucleotide polymorphisms, in yellow are indels, and then blue just adds the yellow and the red together. And when we had Guppy, these errors are too high for doing genetic relatedness. You got to get less than about 20 SNPs. And look what happens when we went to Dorado. They almost disappeared. Median was less than 1 SNP per genome. So just really remarkable and statistically significant. The average Qscore went from 50.5 for Guppy to 60 for Dorado. Again, 1 error per 1 million base pairs. Some of our bacteria are, say, 2 million base pairs, so they might have 1 or 2 per entire genome. All right, here's a little bit of detail. Again, in panel A, I'm just showing the number of SNPs and the number of indels, and they're stratified by the strains. You can see we did have 1 outlier strain. We had a Klebsiella pneumoniae that had a lot of SNPs, but everything else is clustering well below that 20 threshold. What were the SNPs? They were almost all transversions, T to Gs, T to Cs or A to Gs. And in panel C, I show an example. On the top part of panel C is basically the Illumina, which is all calling a G. At the bottom is the ONT individual reads aligned to the ONT assembly, where we're seeing a mixture of A's and G's. The assembly called an A, and so that was an error. But the truth was in there if you could have gotten it right. Being a person trying to run a lab on a limited budget, I asked how low can you go in the sequencing and still maintain its accuracy. So what she did is she took the Illumina reads and said, if I make an assembly out of 20x depth, 40x depth, 60x depth, et cetera, when do those errors basically start to wash out? And the answer was at 20x, it was too low. We saw a significant amount of errors, but by the time we got to 40x, basically the errors disappeared, and at 60, 80, really no difference. So about 40x was our cutoff for getting really accurate genomes, basically sufficiently accurate to assess transmission. And again, the average SNP, again, at just any sequencing depth, was about 1 per genome. Okay, so that's all the sequencing data. Now moving into clinical stuff, how did we assess the potential transmission? Okay, a little bit less important for the purpose of this talk. I think that really how accurate was it and could it be done was more critical. But she used a two-step method. The first is a sensitive method called Core Genome MLST, where you're looking at all the genes that are present in all strains of a particular species. We have a lot of intra-species variation. And for that, you have to have basically, depending on the organism, less than 15 or less than 20 SNPs. And then if they met that criteria, you go on to a reference-based calling alignment where you're looking at the entire genome now, not just at the genes. And again, you have to stay below 15 to 20 SNPs. So you can see that in order to do that kind of accuracy, Guppy would not give us the kind of error rate necessary, but Dorado definitely did. So we were able to confidently call potential transmission. Okay, here's the data. Again, I know most of you guys are not microbiologists, but the general pathogen breakdown was what we would expect. For example, for Klebsiella pneumoniae, there's a highly lethal strain called ST258 that we saw about 15% of the time. Similar for E. coli, a lot of our strains were ST131. And I do want to call attention to VRE. More than 60% of our strains were ST117, which is an emerging clone. We also had a clone of Pseudomonas aeruginosa called ST633. This just means sequence type, and it could be emerging. And the MRSA strains are those that we see in the community. Okay, so this is the data in terms of transmission. Basically what I'm showing here is a circle. Each slot represents one strain, not how closely they're related, but just what the color is. When they're linked by a line, that means they met both of our criteria, both the sensitive criteria and the specific criteria. So we had 242 isolates from 218 unique patients, so a patient could contribute more than one isolate. We had 5 clusters, around 9%, not too far off from the Pittsburgh data. The cluster size ranged from 2 to 12. So for example, there were 2 Klebsiella pneumoniae that differed by 8 SNPs. There were 2 VREs differing by 5 SNPs. And then that huge cluster revolves the ST117 VREs that had about a median pairwise symptoms of 12, but as little as 1 or 2 SNPs. Okay, so how do we integrate that with genetic data? So on the left side of the panel is an individual patient, so it's patient A, B, C, D, E, and the color is the ward in which they're staying, and the time is at the day of the study on the x-axis. Okay, so the black dots represent when they got their infection, and so you can ask the question, okay, were they overlapping in where they stayed? So a possible transmission would be here, panel F and G, because the 2 patients had been on the same same ward, but they didn't overlap entirely in where they stayed. Whereas panel patients D and E would be classified as likely transmission because they were sharing the same ward when they developed this infection. So 15 of the 21 strains that we classified as potentially transmitted actually had epidemiologic links for transmission, indicating that we were picking up likely transmission going on, again, undetected under the radar of the infection prevention and control persons in our hospital. Okay, so the conclusions from this data, I really thought this was a remarkable data that Chen-Ting generated, that she could use Oxford Nanopore sequencing to efficiently and accurately assess genetic relatedness among major antimicrobial resistant pathogens. The strength of the study, it was prospective. We did relatively real-time sequencing. She did it once a week. I didn't bludgeon her to do it 2 or 3 times a week, which would probably be necessary. It was very low resource utilization. Single graduate student doing this outside of a sequencing facility. Weakness of the study, fairly low number of samples. Remember the Pittsburgh data, 3,000 samples, we only did about 250. However, if we added more, we probably would have picked up more transmission, increased our power. Again, we did not try to perform sequencing more than once weekly, and we did not do any interventions yet based on these data. Finally, we did not try and assess C. difficile, which is another AMR pathogen often in the hospital. It's supposedly 1 or 2 SNPs to identify transmission. So again, since we were at 1 per genome, I'm not 100% sure we can use that to do C. diff. So our future directions, given the ease and accuracy of the sequencing, we would like to create an on-demand platform to rapidly integrate into IPC efforts. Really important is ruling out transmission and how we can not have to spend so much time or effort on pseudo outbreaks. We also are interested in drug resistance, as well as helping the clinical microbiology lab with unusual pathogen identification. This is a picture of Chen Ting getting her master's degree from Dr. Spallone, who's the head of infection control, really critical people in this project. And then William Shropshire, who is the bioinformatics person. I have to acknowledge he had a T32 from the NIH that supported his work. And here is my last slide that I was asked to put up. And I want to thank you all for your attention.